Thank you, Dr. Schwartz, and thank you to all of you for the opportunity to come and to present to this great group. It's great to be here and to talk about some of the important issues in the field and to present some of the new work that we've been doing in the past few years. By way of disclosures, you can see them listed there, including that I'm on the supervisory board of QIAGEN. So what I thought I'd do today is start broad and then get into specific vignettes using a disease that I think a lot about in my laboratory, acute myeloid leukemia, as specific examples of some of the things that we've learned in the past few years. But before we drill down into the specifics, I thought it would be important to take an overview and ask ourselves broadly within the perspective of molecular profiling and clinical medicine, where we are and where we're going, and what are the questions that we need to address to get further within the field. About 3 years ago, we really had a salient and critical moment in the field when our former president, Obama, launched the Precision Medicine Initiative and really announced what he believed was the beginning of the modern era, where we begin to use technologies to really match patients with specific therapies based on specific alterations in their health and disease state. And this young woman with him, Alana Simon, was a woman who had a rare liver tumor, who then did whole genome sequencing of a whole set of people with the exact same tumor she had and found a canonical fusion, which had not been previously been found through whole genome sequencing, a great example of how one can use the power of genomics to find things we didn't even know about before. So the goal, I think, for all of us as a field is to ask, how can we use these exciting technologies that continue to expand in power, reduce in cost, increase in availability to impact human health and disease? But if we take a step back and ask ourselves a fundamental question, which is precision medicine a new era? I would posit that all of us know this, but it's not often discussed, and that is that every day at every patient interaction, in every lab test we run, in every radiographic test that we analyze, we make patient-specific decisions. And we use that information to make decisions about how to proceed in clinical care. If we then ask, well, we acknowledge that, but molecular profiling is the new era. I would posit that cytogenetic alterations have been used for diagnosis and for therapy of, for example, leukemias for decades. So again, if we just deleted the word mutational profiling and inserted cytogenetic alterations, we've been doing this for a long time. And there's nothing new except for the use of better and cheaper technologies to apply patient-specific alterations. So I would say precision medicine has been a reality of everyday clinical care forever. And that there's nothing new. The difference is the amount of data we can obtain for each patient, the cost of that data, and our ability to sift through that data presents new challenges. So it's just a more interesting and more complicated endeavor, but the basic concept remains the same. And if we also ask, where are we with respect to that data, it's being accumulated at a tremendous rate. And this is just a few examples from the ICGC, which has been one of the international consortia that have assembled exome and genome data. And you can look at this information all the time. But the number of genomes that have been done for cancer is awe-inspiring. And now as we begin to look at the UK whole genome efforts, and now the All of Us effort in the United States, as well as efforts around the world, we're going to end up probably within the next 5 to 10 years with more than 1 million genomes, both in health and disease, and with cancer being, if not the primary disease where we have the most data, certainly with cardiovascular disease in the top 2. And you can do things like this. This is data from Sloan Kettering using our CBio portal, where we've been able to assemble, thanks to Nikki Schultz and his team, a tremendous amount of data from public consortia. And you can ask about your favorite gene in every cancer. You can drill down at it. And I think one of the exciting things is not just the ability to have this data available, but also to have the ability to mine it and use it on a day-to-day basis. And what I'm excited about when I go into my laboratory is that all of the postdocs and students and technicians have the ability to have this data at their fingertips. And I think that's what's exciting. And the same is true in the clinic, that when I see oncology fellows, they use these resources every single day. And then if we take a step back and ask, how do we then take this information and apply it to patients, there's many examples of success. On the left, this is an example from our colleagues at Dana-Farber that have developed a rapid DNA sequencing panel for myeloid malignancies. And they've done a tremendous job with rapid turnaround time within just a few days, roughly 4 days. And it's available at a tertiary medical center. And that's a great example of an incredibly accurate and useful test that's available in a large medical center. On the right is the example of Foundation One Heme. It's just an example of a test, and we actually helped them design and develop this test that you can do both DNA and RNA sequencing for all of hematologic malignancies, let alone their Foundation One test, which was available for solid tumors. And the important thing here was that technologies like this now make it available to everybody everywhere. Not that there aren't issues about how to deal with that, and I'll come back to issues about interpretation and reimbursement, but this is an important thing that now these technologies are available to patients everywhere. They don't just need to be at a tertiary or quaternary medical center. And so the question I think we have to ask ourselves before we drill down to the specifics is, where are we as a field? How do we go from tremendous power of discovery genomics to clinically using this information every single day in all of our patients? What are the limitations? Obviously, the first would be, are we getting more and more data? The answer is absolutely. So it's not a lack of information, although whether we have the right information, I'll come back to. We have constant rapid improvements in sequencing technology and analysis. We hear a lot about how sequencing gets cheaper. I'll remind you that we have this dueling thing where sequencing costs go down, but analytic requirements go up. So it's not entirely clear if overall costs are going to continue to go down. We have the expansion of clinical-grade genomic platforms, both in academic centers and in central laboratories, and I think ultimately around the world. But if we take a step back and ask ourselves, there's still the majority of patients in the United States don't get genomic profiling as part of the routine clinical care. So we have to ask ourselves why that is. And I'll just remind everybody, and I'll come back to reimbursement later, that, as someone who takes care of cancer patients when I'm on the wards, the cost of a genomic test, even with the appropriate inflation and thinking about the delta, what you get reimbursed versus what you say it is a minute fraction of the care, of the cost of all of our patients. If we look at the radiographic tests we get, the cost of every night in the hospital, the drugs we give. So the idea that genomics is going to break the financial bank within respect is just not true. I think it's just that the sensibilities around a new modality being brought into care where we don't have an established reimbursement structure creates realities that we need to deal with as a field. So what I thought I would do today is focus on one disease that we've thought a lot about and illustrate some concepts, both with respect to clinical profiling and also respect to biology and what we've learned, and try to address these questions in the next 40 minutes or so. Acute myeloid leukemia is a disease that I, as a clinician and scientist, have thought a lot about, and many of you think a lot about in your laboratories all the time, because it's really been the disease where cytogenetic profiling has really been the cornerstone, both for this disease but more broadly. But I'll remind you that until about 2 years ago, we really had very little improvement in outcomes in AML. Improvements in antibiotic care, supportive care had led to marginal improvements. We'd had no new drugs approved until about 2 years ago in 3 decades, with the exception of hypomethylating agents. We now have new drugs, and I'll come back to that, and we're excited. But it's a disease where, despite tremendous interest, tremendous science, we had not really translated that to the clinic. And that really was why I decided to focus on this disease a number of years ago. And so one of the questions one has to ask is, why was AML so hard to decipher? What made AML such a difficult disease? So I'm going to give you the glass half empty, and then I'll give you the glass half full perspective. The glass half empty perspective is shown by this gene expression classification of AML. And if you look on the left, you can see 16 groups. These are robust, they're reproducible. This was arrays. You can do this by NGS, RNA-seq. The lesson here is that AML is not one disease. It's probably about 16, at least in transcriptional space. And that is very different, for example, than many adult epithelial tumors where you can have 4 or 5 distinct groups. So you have a rarer disease and much more complexity where patients can be in different bins. Imagine doing a clinical trial of 20 patients with a new drug and you treated and you actually the drug only worked in one of those groups. You're very likely to miss that signal. So that's been the challenge we've had in AML. If we look at epigenetic space, this is just methylation profiling of the exact same cohort of patients from the previous slide. You get a similar clustering, but the lesson here is that not all these groups read out in transcriptional space. And people struggle with this. Why do gene expression and methylation not read out the same thing? I'll remind you that transcription reads out what a cell is doing, and methylation reads out what a cell can do. They're not reading out the same thing. It's about steady state versus capacity. So one might imagine that when you combine them together, you actually get orthogonal information. So there may be as many as 20 or 22 distinct groups, some of which are read out by transcription, some by methylation, and some by both. So that's the glass half empty perspective, that AML is just complicated and we're never going to figure it out. The glass half full perspective would be this slide. This is data from TCGA looking at the genomics of AML. And the lesson here would be when one looks at somatic coding mutations, you only find 3 to 8 coding mutations per AML genome. And they only occur if you really ask about 1% or greater in about 50 genes. So what that means is that you get tremendous biological diversity, but from a relatively small number of genetic events. And what we believe is that the small number of events generate enough permutations with the different cooperation and combinations of mutations where you can generate that biological diversity. But this is very different than melanoma, for example, where with a UV-induced mutagenesis signature, people spend hours and hours and years, arguing about what's a statistically important driver versus a passenger, and where are the real genes once you get outside BRAF. Here, we know that these 50 genes are important, and we know that they generate diversity. And so we can at least take a stab at trying to understand the question both from a genetic perspective and from a biologic perspective. And so what are the questions that I want to address today in my talk? The first would be, what has genomic profiling taught us about the biology of this disease? What are we learning from genomic profiling of patients, and both about disease initiation and progression? The second would be, how can we use genomic profiling to improve clinical care in AML and other malignancies? Have these insights led to novel therapeutics, and what are we learning about novel therapies? And finally, can we use all of this together to accelerate translation to the clinic? And can we change the paradigm and actually develop a much better model to get patients to the best drugs and get drugs to our patients as an approved therapy faster? So what I thought I would do today is tell one brief vignette of sort of an interesting story that we've been working on for many years and what's become an exciting new field. And that focuses on a set of mutations that we and others have been working on for the last few years. And these are mutations that are in the enzymes that regulate DNA themselves through chemical modifications. We know that DNA can be methylated, starting with cytosine, and that DNMTs, including DNMT3A, which is mutated in AML, can place a methyl group. And we've learned in the last few years that the TET enzymes can place a hydroxyl, a formyl, and a carboxyl group on methylated DNA, and that leads to demethylation in DNA. And that's something we didn't even know about 10 years ago, but in the last 10 years, we've begun to understand. And what you get in AML is either through loss of function TET mutations, which then block those steps, you'll accumulate methylcytosine and deplete these other steps and end up with a hypermethylation phenotype. We also showed, working with Ari Melnick and Craig Thompson, that IDH mutations produce this metabolite, 2-hydroxyglutarate, and that poisons the TET enzymes directly. So loss of TET or gain of IDH results in blockage of those enzymatic steps. So this has been, an exciting biological observation. I'll come back to therapeutically targeting IDH in the last few minutes. But what I want to talk about is fundamentally is how do these mutations that are on this slide, DNMT3A, TET, IDH, contribute to transformation? And what can we learn about their role in the biology? Because we really knew nothing about them until the past few years. To do that, I have to tell you a little bit of a historical story that really, to us, is one of the more exciting things that has happened in the field in the last few years. And that is this concept of clonal hematopoiesis. In the 70s, as many of you know, Fialco used G6PD polymorphisms on the X chromosome to show that chronic myeloid leukemia and other leukemias were clonal in women that were polymorphic, because X inactivation is stochastic and is inherited after lionization in every cell. So you can use it as a surrogate for clonality. About 30 years ago, Lambert Busk, who was the first postdoc with Gary Gilliland, who is my former postdoctoral mentor, now the president of the Fred Hutch, used a PCR-based assay that Bert Vogelstein had developed for colon cancer to look at clonality of leukemias. And they were using non-random X inactivation to look at whether different types of leukemias were clonal versus not. But Lambert is a really, really thoughtful and rigorous scientist. He's a leukemia doctor and scientist in Quebec at the University of Montreal. And what he decided to do was to do age-matched normal controls that didn't have leukemia and ask, do I see clonal alterations in the absence of leukemia? And he absolutely did. And what he observed was an age-dependent increase in these clonal outgrowths of hematopoietic cells. And so he argued, now about 30 years ago, there was a phenomenon he called clonal hematopoiesis that led to the expansion of pre-malignant cells, and perhaps these might be a reservoir for subsequent leukemic transformation. Now, this led to a huge debate in the 90s and 2000s about whether it existed. And the reason I got involved, actually, was that Lambert and Gary were involved in a back and forth in the journal Blood about whether clonal hematopoiesis existed, whether this assay was accurate. And Gary had actually gone to Merck, and Lambert is French-Canadian, and he asked me to be a co-author on the letters because he needed an American co-author to edit his language. And so we were sitting at the bar at ASH in 2009, and we were talking about this, and I said, you know what? Exome sequencing is now available as a new technology. This is a perfect experiment. Take some of these women that have skewed X chromosomes, take their blood, take their epithelial DNA, and if we find mutations that are only in their blood and not in their epithelial DNA, we'll have proven that clonal hematopoiesis exists. And that's exactly what we observed, were somatic tattoo mutations in women that did not have leukemia, that had clonal outgrowth. Importantly, these mutations were easily detectable by Sanger sequencing, so these are not in 1% of their blood cells. These are in 20, 30, 40, 50% of their blood cells in people that don't have leukemia. And the mutations we observed were functionally known to inactivate the TET protein. Either they were nonsense frame shift or they were missense mutations that catalytically were dead. So we had no doubt. And in fact, what we found was that if you look in normal folks over the age of 70, that about 2% of people over the age of 70 have TET2 mutations. And that means that if we go out to the street here and take elderly folks and we sequence 50, we're likely to find one. And what we found was that the frequency of TET2 mutation leukemia is about 8%, especially over 70. Maybe it's 10 or 12%. So that means that these mutations are occurring in an age-dependent phenomenon, both in normal individuals and in leukemia. And that really opened our eyes to this idea. So that's sort of where we were when we described this observation now about 6.5 years ago. Fast forward to 2018, this is now a field. And what we've learned from many different folks in the field is a lot of fascinating observations. Ravi Majeti at Stanford, as well as John Dick, took leukemia patients and were able to fractionate immunophenotypically normal cells from people that had leukemia, so they could flow sort blasts versus normal stem cells. And what they showed is that some of the normal stem cells had these early mutations, but not the later leukemic mutations. So even in a patient with leukemia, you can have residual clonal hematopoiesis. And if you treat those people, the leukemia cells may respond, but not always the pre-malignant cells. We know from population-based studies from TCGA, as well as from Ben Ebert and Steve McCarroll, that if we now take unselected analysis of normal, healthy individuals that don't have any evidence of cancer, or in the case of TCGA, who have solid tumors, that about 10% of people over the age of 70 have clonal hematopoiesis with known leukemia drivers with mutations in genes like DNMT3A, TET2, ASXL1. I'll point out there was just a paper from Steve McCarroll and colleagues in Nature about 3 weeks ago that CNVs may be as, if not more common, so that if you then include copy number alterations, you may get up to 30% of all people over the age of 70 have evidence of a clone that can be measured. We know that the implications of this are just beginning to be understood. First of all, if you have people with solid tumors and use blood as your reference, you might identify leukemia mutations in their blood. I'm going to talk about that in detail. If you're following a minimal residual disease, how do you deconvolute a mutation that might not be only in the transformed clone, but in these pre-malignant or antecedent clones? And Ben Ebert and Kevin Walsh have shown both in mouse and man that the risk of cardiovascular disease if you have clonal hematopoiesis is similar to having an aberrant cholesterol. So that means that this is not only relevant to cancer, but probably to almost every disease of aging. And what we believe fundamentally is that in 5 years, we're going to look back and say, it was amazing that we described this phenomenon, and now we're going to begin identifying and treating it. And we believe this may be the first example where now, using molecular profiling, we can identify people at risk for getting leukemia and treat them before they have leukemia. So that's where we think the field is going to go. And so our model for how leukemia develops really is shown in this slide. That leukemia begins with early mutations that don't cause disease by themselves, but increase the fitness of hematopoietic stem cells and allow the stem cells that have those alterations to outcompete and outsurvive their colleagues. And that there are other events, mutations that we do and don't fully understand, that then give rise to progression to an overt disease. And if it's a FLT3 ITD, it'll be AML. If it's a splicing mutation, it'll be MDS. If it's a JAK2 mutation, it'll be a myeloproliferative disorder. And what we don't understand is whether once you have a transformed disease, is the best therapeutic target the mutations that cause the cells to go over the barrier for transformation, or can you target those early events both in a pre-malignant or malignant context? So these are the questions that we and many others in the field are trying to address. But what I thought I would do is talk a little bit about the implications of clonal hematopoiesis before I come back to AML, because I think there's still a lot we're trying to understand. So this was a first study we did with my colleague Larry Norton, who's one of our icons in breast cancer therapy at Sloan Kettering. We took a small number of young women undergoing surgical resection for breast cancer. So these were pre-therapy, so no chemo, no radiation. And we flow sorted from within their tumor, their white cells, and we could show that in 10 of these 15 women, we found somatic mutations in their leukemia cells, and they often were in known leukemia drivers. And the women who had these mutations had an increase in pathologic leukocyte infiltrates, particularly myeloid cells, suggesting the idea that this was common, was easily detectable in people with solid tumors, and might have biological relevance. And then this is another example of a patient that we saw at Sloan Kettering. This is a woman who had endometrial cancer who showed up to Sloan Kettering with her Foundation Medicine report. We see this all the time. She had a number of mutations you'd expect with endometrial cancer, like KRAS or PIK3CA, but she also had a DNMT3A mutation. And if you look at this report, which is a tumor-only sequencing platform, it suggests perhaps using hypomethylating agents for DNMT3A mutant endometrial cancer, which is a bit of a stretch, but it's possible that it might work in other contexts. The problem, of course, was that it wasn't in the tumor. And so one of the things we did for this woman was resequence her tumor and her white cells. And what we could show is that the RAS mutation, which could have actually been in either clone, if you think about it, RAS is both a leukemia and a solid tumor driver, is in the tumor but not in the blood. But the DNMT3A mutation is threefold more in the blood than in the tumor, and this is almost certainly admixed white cells within her tumor. So this suggests the idea that perhaps with tumor-only sequencing, we're vastly underestimating the contribution of clonal hematopoiesis to false positives. And the second is the clinical relevance of this was not well understood, and so we thought this was an opportunity to study this phenomenon. So clonal hematopoiesis is common with aging, so are all of solid tumors. We don't know if these mutations are relevant to solid tumor biology. We also don't know if people get systemic treatment for their cancer or radiation therapy or surgery or anything else. Does it affect these clones? And we also didn't know at the time whether secondary leukemias would develop in the people that had these mutations versus those that don't. Could you use the presence of mutations at the time of solid tumor diagnosis or during therapy to predict who's at risk for secondary leukemia versus those that are not? And so this is where we were very fortunate to work with our solid tumor colleagues at Sloan Kettering, who developed the MSK Impact assay. And that was that assay I showed you a few slides ago. It's a tumor versus reference targeted sequencing of 410 genes. We've now done about 28,000 patients. And what we did was a couple things. The first is because all of the reference, or germline, if you will, is blood, we were able to turn the analysis on its head and ask what's in the blood that was more abundant versus in the tumor. So ask the reverse question. The other thing was that we built into the consent all patients signed that we're going to look at your blood for these mutations, and we want to be able to link back to you. So we built into this consent now about 3.5 years ago the idea that we could link back. So all of this is identified research. It's not de-identified. And then we did brute force analysis of charts. There is no heuristic to do detailed analysis of clinical parameters. So we had residents and medical students who mined every chart. And we looked at initially at the first 8,800 patients in the first manuscript, and we're now analyzing the full 28,000. So the first thing we observed was that when you look at Sloan Kettering, 25% of all solid tumor patients who get sequenced have clonal hematopoiesis, and the mutational spectrum is very similar to what we observe in the normal population that don't have cancer. The frequency of CH is more common in our cohort than in previous cohorts. These are those other cohorts I talked about before, and you can look that in our cohort, once you get up to about age 75, you're about 50-50. So that's pretty amazing that half of all cancer patients who are 75 have a leukemia mutation in their blood. So of course that leads to an interesting question. Is that because cancer patients who are being treated and have solid tumors have more mutations, or is it because we're doing higher depth tumor normal sequencing? We're not looking for hotspots. We're calling every mutation through paired analysis. And so now we're collaborating with the groups in Boston to determine how much of this is the difference in population versus the difference in analysis. And that's something we'll report on in the next year. Most patients have just one mutation, but we've seen as many as 11. And one of the things that we're doing now in the lab is collaborating with our colleagues at Mission Bio to use single-cell analysis to determine whether these mutations are in the same clone or whether a patient who's got 3, 4, 5 mutations has multiple clones. Because either way, it's interesting, but it's important to know that having multiple mutations, is that a flag for clonal evolution of a single clone or indicative of multiple clones popping up? We just don't know the answer based on this genomic analysis. And we flag all patients with more than one mutation or have a 10% VAF or greater for clinical follow-up, which I'll come back to. And what we found is that if we sort of blinded our eye to the blood sequencing and said, well, what if we only did tumor sequencing, like using many of the assays out there, 5% of all patients would be miscalled as having mutations that one would think are tumor-derived but are actually blood-derived. The problem with this is not the DNMT3As. It's pretty obvious that if you have a DNMT3A or TET mutation, those are likely to be hematopoietic derived. But look at the examples of ideation RAS. What do you do for a gene that can be mutated in a solid tumor or in a liquid tumor? Without tumor and blood sequencing, you can't deconvolute that. You could treat a person with an IDH mutation with tumor sequencing, but you could be treating their leukemic cells that have the IDH mutation versus their tumor, and the same is true for RAS. And so that has very important implications, and so we think it's an argument for doing paired tumor and reference sequencing if one does high depth and extensive NGS. The other thing that we learned from this was, I think, a biological observation that I think is really fascinating. On the top, this is the lollipop plot of all of the mutations in the DNMT3A that we observed in our cohort, and we went down to 0.5% VAF because of our depth. On the bottom, that's the distribution of mutations in DNMT3A and AML in 1,500 patients from my colleague Ellie Pep-Emmanuel. And you can see the distribution's different, with, in fact, a bigger spread of mutations in CH than in AML. However, if you were to restrict this to the mutations that get to 10% or greater, the top lollipop plot looks just like AML. And what that means is, conceptually, that 3A mutations maybe happen for reasons we don't fully understand all the time. But only a subset of those mutations have the right fitness advantage to pop up to 10% or greater. And we think a combination of the wrong mutation in the wrong person who has the wrong stressors, inflammation, obesity, chemotherapy. So it's a combination of the wrong genetic alteration in the wrong host that leads to that popping out. And I'll just point out Todd Druley at WashU has done some really elegant work with bar-coded sequencing showing that he can identify DNMT3A mutations in every single person once you hit 40. So we don't think the rate-limiting step is mutagenesis. It's the wrong mutation in the wrong host, and then what else happens to the person. And so we think that it's not mutagenic rate per se, but it's mutagenic identity on top of the fitness landscape that affects what mutations occur. So next we asked, does therapy for cancer affect clonal hematopoiesis? And I would have bet every dollar in my pocket that chemotherapy would have been the primary predictor. It turns out it's not. In fact, it didn't survive multivariate analysis. By contrast, radiation therapy, and I'm not talking about mantles. We had no lymphoma patients in our cohort. We excluded all blood cancers. This is localized radiation, radioactive iodine. So radiation therapy to any site increases the risk of clonal hematopoiesis. We've done serial sampling in people who didn't get radiation, got radiation. It increases the variant allele frequency over time. So radiation is somehow a stressor that can change that. And the other was tobacco, and I was fascinated with that. No one ever thinks of tobacco and leukemia. It turned out I went back to the literature. There's never been a negative study looking at tobacco and leukemia. Every study ever done is positive. It's just lung cancer. It's such a major scourge on our society that we don't think of leukemia, but leukemia is a major result of tobacco use. We then asked about overall survival, and so on the left, in all patients with CH, and on the right, those that have 10% or greater, we saw an impairment in overall survival, particularly in those 10% or greater. It survives multivariate analysis, including age adjustment. But the cause of death here is not leukemia. This is only 18 months to 24 months follow-up. These people are dying of their solid tumor, suggesting potentially, as you heard just a few minutes ago about monocytes in solid tumors, that these cells somehow are interacting with the tumor cells, and so cell non-autonomous mechanisms that contribute to disease. So that was one of the interesting surprises. But what about therapy-induced leukemia? So in the red curve, this is therapy-induced leukemia in the people that have mutations. And on the black curve, both on the left and the right, depending on whether you say all CH mutations versus the ones that have big clones, those that don't have mutations. You can see that virtually every therapy-related leukemia in our cohort at this early point in follow-up occurs in someone that had a mutation previously. So imagine a patient with breast cancer who is deciding with their 1 centimeter node-negative hormone receptor-positive tumor to get AC dose-dense Taxol or just to get hormone therapy. Imagine if they had a large clone, how their risk of leukemia now might be 2 or 3%. The absolute risk of leukemia at 2 years is 2 to 3%. And if we've learned anything, for example, from the Hiroshima and Nagasaki, was that the risk of leukemia is going to go up probably for decades in these people. We are vastly underestimating. I predict we'll see as 10% risk of leukemia in some of these folks over time. So we think the next step will be to get prospective studies or retrospective analysis of large adjuvant studies to determine if we can actually predict, because ultimately it may be relevant to deciding who to give systemic therapy to. And how does this work? I'll just show one functional experiment that we've done. So we wondered, if you think about mutations in blood cells and how they might affect cancer outcome, you have two possibilities. The first would be it makes tumors grow more, invade more, metastasize more. The second possibility is that none of these people in this cohort are being observed. They're being treated, right? So the other possibility is that these mutant cells don't affect the growth of the tumor, but they confer resistance to solid tumor targeted therapies, right? And so we tested this in our breast cancer models, and what we were able to show in this anthracycline responsive model was that mutating TET2 in hematopoietic cells in a breast cancer model doesn't affect the growth of the tumor. However, if you treat a mouse with a wild-type hematopoietic compartment with doxorubicin, the tumor shrinks. But if you treat a mouse that has a mutant hematopoietic compartment, the tumor doesn't shrink, suggesting the effect isn't tumor growth, but therapeutic resistance to systemic therapies. And so we think this is an exciting area to think about the biology. So clonal hematopoiesis is incredibly common. Radiation and smoking are among the most common risk factors. P53 and PPM1D mutations do get selected for by chemotherapy, so I don't want to leave you with the idea that it's just radiation. Unique mutations have unique effects on blood counts. I don't want to get into that today in more detail. I'm happy to talk about that after. And there's effects on outcome that we're just beginning to understand. But what we're most excited about at Sloan Kettering is that we've decided to begin following these patients. So we started a clinic in January of this past year for all patients that we think are at high risk for secondary leukemias. These are our patients with solid tumors. So any patient who has a mutation of 10% or greater or more than one mutation or has a cytopenia plus any mutation gets referred. We bone marrow all those patients. And for now, we're just estimating risk of leukemia, but our vision is to use this clinic as a therapeutic platform for therapeutic intervention studies to try to prevent progression to leukemia. Drugs, lifestyle alterations, exercise, we think anti-inflammatory strategies. A lot of the things that didn't work in the unselected population might work in those that are genetically at highest risk. And so our vision is that clonal hematopoiesis, through inflammation, through cell-cell interactions, through other mutation, acquisition of other mutations, lead to heme malignancies, lead to cardiovascular disease, they affect solid tumors. And I would predict, again, that we'll show in the next 5 to 10 years as a field writ large, that if you look at all diseases of aging, pulmonary fibrosis, kidney, end-stage renal disease, that we're going to find that clonal hematopoiesis is associated with the progression of many other diseases. So the fundamental question will be, can we develop therapeutic interventions to actually ablate these clones? And that's what a lot of us are spending a lot of time thinking about moving forward. And so what I thought I would do for the second half of my talk is flip it from pre-leukemic disease to overt leukemia and give some observations about what we're learning and where we're going. And so how has genomic impacted clinical cancer medicine? And I'll talk about AML, but nothing I'm talking about will be germane just to AML. I'm just going to use that as an example. Obviously, we use it to identify novel targets. I'll come back to IDH in the last few minutes. But can we use it to predict outcome for our patients? And most importantly, even if we don't have a new drug, can we use it to predict the response to existing drugs? And I'll just remind you that there's salient successful examples like 5-q-minus and lenalidomide in MDS and AML, where chromosomal alterations predict for the response to agents where we didn't initially know their mechanism of action. And so we began working on this many years ago and did our first foray into this by doing genomic profiling of a large ECOG cohort. And we initially did just 18 genes in about 500 patients. And what we showed, and this is just 2x2 analysis of mutations, but a better way to look at this is through Circo's plot, that co-occurring mutations don't occur randomly. That specific cooperating mutations occur non-randomly. Particular mutations co-occur with other ones, and that suggests there's interesting biology. But I think the most important thing is that means that you can't say that gene X predicts prognosis or gene Y. You need to say that this combination of genetic alterations predicts outcome this way versus this amalgam of alterations in another patient. And we, in fact, showed in our cohort that we could use a combination of cytogenetics, FLT3 mutational status, and just 8 other genes to refine classification in AML. And this just shows the curves where you can take patients that were on the middle curve and based on additional genomics, either upgrade them to better outcome or downgrade them to lower outcome. One of the problems with this analysis and virtually every other analysis in the field has been that it's done on the same cohort where the predictive schemes are derived. And that, I think, is a challenge. It really limits the robustness of these. So one of the things we did in this analysis was repeat the analysis on a second set of samples. And I think this is a critical bar that we don't do enough in biomarker validation. We need to validate our biomarkers in additional patients. We can't just say gene X predicts outcome. We need to go out and do it again, and do this ideally as on homogeneously treated cohorts. And this, to us, suggested that we could use genomics to prove outcomes. And in fact, 1/4 of all AML patients using this relatively simple genomic testing can be reclassified into unfavorable or favorable based on this. But the best news about this study, which is just 5-6 years old, is that it is totally obsolete. And that is the kind of thing that makes me happy, is when the field goes beyond that to actually take it to the next level. So this is data from Peter Campbell at the Sanger, Hartmut Donor, Ellie Papamanuel, and one of my colleagues at Sloan, where they took 1,540 patients with AML and did 111 genes with targeted sequencing and asked, could they do a better job of classifying? And what they showed were some interesting things, and I'll just give one salient example for the AML aficionados, that if you look at patients that are NPM1 mutant, whether they're DNMT3A wild type or mutant, the FLT3-ITD, it turns out in their cohort, only has prognostic relevance when both these other genes are mutated, but not when only NPM1 is mutated. This is the kind of granularity that you only get from thousands of patients. And we need studies of this size that can be used to predict outcome moving forward. The problem is, once you get to this granularity, is you can't keep it in your head. At least I can't keep it in my head. And so it used to be until 3 or 4 years ago that when my colleagues called me up and they said, I've got a patient X with this mutation or these 2 mutations, I could say, you know what, that's a good or a bad outcome. You just can't do that anymore. So one of the things I think is really cool that they did was develop a specific website where you could actually take your patient's information, not just their genetics, but also all of their clinical parameters, plug them in, and actually get their survival with or without transplant. So I think this is the way things are going to go, that we're going to have to develop heuristics where we take all of the information, not just genomics, cytogenetics, clinical, everything, and develop predictive models. The challenge is they need to be derived on thousands of patients and validated on thousands of patients, or else they're just exploratory. And so we think this will be the future of molecular profiling moving forward. And so where are we going at Sloan Kettering? As I told you, we have MSK Impact, which has now been done with 28,000 patients as of this week. We launched Impact Heme in 2017, and this is our heme-specific version. And so the challenge, of course, is what's the best reference normal for hematopoietic tumors? The answer is there is no best reference normal. So we're using, in this case, fingernails, with a caveat that we're quite aware that this has its limitations and that there is disease contamination of every normal, but you got to do something. Or, perfect is sometimes the enemy of good. And so we believe that doing tumor reference sequencing for leukemia patients, lymphoma patients, and myeloma patients is the way to go. What about regulatory guidelines and reimbursements? I think this is an area where we as a field have been passive, and we have let the regulators futz around too much, and we need to actually be much more proactive about guiding this. There's incredible variability in state-to-state guidelines. I live in New York, where there are tremendous rigor in these guidelines, and we have, I think, more rigorous guidelines than most other states. But why should this be state delineated? Why should these guidelines be different if the test is run in Manhattan versus across the George Washington Bridge in New Jersey? The role of the FDA in regulating these assays is evolving, and I think we need to be very thoughtful about how to do that. And we have 2 options. We can let the FDA figure it out, or we can help the FDA figure it out. And I'll argue that we should help the FDA figure this out. I think in my engagements with them both within the diagnostics branch and the therapeutics branch, they want to be part of it. They don't want to just come from their ivory tower and tell us what to do. And I think that's something we need to do as a field, and this group should be part of that dialogue. And reimbursement, I don't want to get into the whole reimbursement landscape, both within FDA approval and within Medicare and Medicaid, but it's my hope that we're going to have many, many more genomic tests that are widely reimbursed in the next 3 to 5 years, and that this will not be a major limitation to patients getting testing, and it remains for many of the assays we want. And then the last thing, which I think is really important, which is education. One of the things that I really am heartened by in my clinical practice and in my role at Sloan Kettering is that the oncology trainees, the pathology trainees we see are living, breathing, and eating this stuff. They know it. They read it. They know it. They learned it in medical school, and they do it all the time. And for them, it's going to be like riding a bike. They're going to pick it up all the time. But that's just one group. What do we do with established physicians that are 60 years old that don't even understand how this is all done? How do we educate them? What do we do for non-physician healthcare providers, for nurses, for nurse practitioners, for physician assistants, often whom are the person answering the patient's question? I have this genomic result and I don't know what it means. And what do we do for patients and families? So what I believe is that we need to take this head on. So trainees need to learn genomics. So one of the things that I really am pushing for within hematology, the same is true for oncology, but I, because of my role in ASH, have more of an ability to impact this. I think that hematologists in training at all of our institutions need to rotate with all of you in genomic analysis and molecular genetic analysis. They need to do a rotation the same way many of them do a hematopathology rotation to learn morphology. They need to learn molecular profiling. And the same is true for hematopathology. I don't think you can be a hematopathologist and not understand this. Even if you say, you know what, I'm going to be more of a pathologist than a molecular hematopathologist than a molecular pathologist, it would be ridiculous for folks to go into that field and not have this training. And many of these fellows are going to include this in their scholarly research, whether in the lab or the clinic, and we need to enable that. And we need to incorporate this genomics into CME and MOC for physicians and other healthcare providers. So I think this is on us as a group to figure out moving forward. And so in the last few minutes, I thought I would talk a little bit about how do we use this information for therapeutics. Again, the first examples of targeted therapies based on specific genetic lesions emanated from hematopoietic malignancies, BCR-ABL being the most salient example. But most recent progress in clinical development has come from solid tumors. For example, non-small cell lung cancer and the litany of kinase mutations and fusions, which lead to drug sensitization. There have been many more precision medicine trials that are solid tumor-based. So how do we move forward and get this to work better? How do we use genomic data to accelerate therapeutic development? How do we use that information to do this? And again, as a hematologist, I would like to see the blood cancer field be a leader and not a follower in this area. And so where are we within AML? From my standpoint, we're at a sort of pivotal moment. We now have drugs that are starting to show targeted activity, FLT3 inhibitors, IDH inhibitors, which I'll talk about in a minute, spliceosome inhibitors are entering the clinic. We're beginning to see clinical benefit, but these just begin the journey. We don't know how these drugs work and who they work in and who they won't work in. We don't know how other genetic alterations in these patients affect the response to these drugs. We don't know how to combine these drugs with other therapies in AML. And we don't know how to do the right clinical trial to really get these drugs to our patients quickly. And so I thought I'd give one example with IDH2, and then I'll end with a novel clinical trial. So we've been very involved in the IDH2 story. This is work done in my lab with Agios and Celgene, where we showed that the IDH2 inhibitor in our preclinical models could block the long-term capacity of IDH mutant leukemia cells to propagate. I'll point out that in short-term assays, they expand because of the differentiation effect. So if you did a one-week assay, you'd actually score this as promoting the growth of leukemia. You have to culture them for 6 weeks. And if you take other leukemias like TET2, nothing happens. Based on that work, we went into the clinic. This drug, the first drug, IDH2 inhibitor, was approved in August 2017. The IDH1 inhibitor was approved about 3 weeks ago. And we see evidence of differentiation and a response rate of about 40% with a complete response rate of 20% in relapsed refractory leukemia. That's great. But treating a targeted agent and only getting 20% complete responses is a partial success. So that leads to many interesting and important questions. The first would be, how does the drug work? One of the fascinating things about this drug is that when patients are in remission and we take their neutrophils at the time of remission and sort them, these neutrophils have all the mutations for the pre-therapy blasts. So this drug isn't ablating the molecular clone. It's differentiating it and making it look normal. But those cells that look normal flow cytometrically are molecularly clonal and abnormal. And that's a different mechanism of action than a kinase inhibitor. And that, to us, is very intriguing. And that leads to a lot of interesting questions, which would be, first of all, do other mutations impact that response? Why is only 20%? Well, how do patients, when they respond, then relapse? And can we use these insights to develop novel combination strategies? So I thought I would just give one example of each of those ideas. The first thing we did was ask, does co-occurring mutations that are at the time of diagnosis or at the time of therapy predict for response or lack of response? And what we've found so far is that, now this number is about 30 patients, is that of the 30 patients we've treated that have a RAS pathway mutation and an IDH mutation, only 2 have responded. So if you have a RAS, KRAS, NRAS, or PTPN11 mutation, your chance of responding to an IDH inhibitor is almost 0. So that means it's not just IDH that predicts response, it's IDH with or without RAS. And the biology of that, you have to give us some time and others to figure out. And that, to us, is very intriguing. The other question would be, when patients are treated with the IDH inhibitor and they respond, if they relapse, what's going on? And again, these are new agents. These agents promote differentiation. So one might imagine that resistance might emerge in different ways. So we've studied a number of patients that had IDH inhibitor responses and then relapsed. This is a patient who got the IDH inhibitor. Their blasts went down. Their neutrophils went up because of the indifferentiation effect. But then they relapsed. And when they relapsed, the 2-hydroxyglutarate, which is the product of the IDH enzyme, came back up. So we hypothesized this wasn't due to some other genetic event, but was because the IDH mutation was restoring its activity. We identified another patient just like that. And in both patients, we identified other mutations in IDH that were not seen pre-therapy, that were actually far away from the IDH mutation that's activating. So that to us was exciting, that you could find other mutations in IDH at the time of relapse. We did digital droplet. We cannot see these mutations pre-therapy. So we looked at the crystal structure of IDH2 bound to the drug. And what we found when we showed this to our colleagues, because the crystal structure had already been solved, was that they were obvious. The mutations we observed were at 316 and 319. They're exactly where the drug binds. It wasn't surprising. But there was a wrinkle. It turns out that the IDH enzyme is a dimer. And you have a mutant part of the dimer and a wild type half of the dimer. And so that leads to the intriguing notion these mutations could be either in the mutant allele, in cis with the activating mutation, or in trans, in the other copy of IDH, which had not been described for a targeted therapy. And what we did was take these patients, because a few kilobases away, and just isolate large fragments of genomic DNA and clone them and sequence them and ask, are they in cis or in trans? And in each case, these mutations were in trans. So these mutations that cause resistance are occurring in the other IDH allele. And that's a novel mechanism of resistance to targeted therapy, although I would argue that probably it's never been studied. And if you actually looked at this and did careful allele-specific analysis, some of the other examples of resistance might be similar in mechanism. We went into mouse models and showed that this results in restoration of 2-HG production in mice. Of course, we already knew that from people, so it's not that surprising result. And then we went to our colleagues at Agios and said, we've got this interesting finding with IDH2. And they said, it's funny, we have this patient on the IDH1 inhibitor trial. They developed resistance, and they have 2-HG going up, and they have a mutation at serine 2080. And when we lined up serine 280, it lined up exactly where the mutation in IDH2 that we described at isoleucine 319. And what we were able to show in this patient was that this mutation was in cis. So what that means is that the mutations can occur in IDH1 or in IDH2 in cis or in trans. And that, to us, suggests that you can do genomic analysis of these people on trials and identify new mechanisms. And if you pay attention, find some really interesting new wrinkles. So a subset of patients on IDH inhibitor therapy develop resistance. Some of them have 2-HG come back up. This is associated with the acquisition of second site mutations. They can occur in trans or in cis or with IDH1 or IDH2. And this further credentials IDH enzymes as a therapeutic target in AML, and we hope in other diseases. So that really, from our standpoint, is an salient example. Epigenetic targets are relevant to AML, to the initiation and maintenance of disease. They co-occur with other mutations like RAS that affect the response to these drugs and affect transformation and progression. They have novel mechanisms of activity, and we can identify by careful analysis of patients how these drugs work and how they don't. But the fundamental question, of course, is how do we take all these insights of new drugs and new targets and new platforms and get this to the clinical setting? And so one of the things that we were really struggling with in the AML field was that there was no ability to take patients' genomic information and to line them up to the best therapy, especially in the upfront setting. And so about 3 years ago, a group of us, specifically John Bird, Brian Drucker, myself, and Amy Bird at LLS, decided that we would launch a new trial where we tested this idea of precision medicine and AML, and that we would get everyone to work together. This trial had some unique angles, including the fact that the LLS, as a foundation, holds all the INDs and took medical legal risk for this trial. They did all the fundraising, and then they got all of the academic institutions as stakeholders, as well as the pharmaceutical companies to pitch in and say, we're going to work together. And the caveat we said was that we're going to all share credit on this trial. This is the design of the trial. The only eligibility criteria is you have to have AML and you have to be older than 60, and you have to be newly diagnosed because we want to give this as frontline therapy. We sequence all patients within 7 days. They get both cytogenetic as well as molecular genetic analysis. We're using Foundation Medicine because this was the FDA-approved IDE assay for the trial, and we have to get the data back in 7 days or less. Patients are waiting with AML for treatment. If you have a marker that predicts therapy, you get a targeted agent. And if you don't, we can give you, for example, an antibody against the cell surface like CD33. So everyone on this trial gets a new agent. What's exciting about this trial is we're not giving these patients intensive therapy. We're giving them novel therapies as first line. People can always say, you know what, I want to get chemotherapy, but for older adults, the outcomes with chemotherapy aren't great. And so what we're excited about in this trial is the idea that we can test new therapies in genetically enriched populations that are likely to respond as their first therapy. And we're going to test 30 to 40 per arm and look for big response rates, 40, 50%. And if we get that, great, we'll move forward to registration. If we don't, I think ultimately if somebody wants to pursue a 10% response rate, that's not where we're going to go in this trial. So this trial is going great. We're doubling our projected accrual. We're at about 260 in the first 16 months. We were hoping to get to 500 in 5 years. We've got these sites. You can see them listed up there. I think it's now 11. That includes 3 Mayo sites, all 3. And we're intending to go to at least 15. We have 6 arms currently, but we're intending to have 10. And these arms open and close. They're dynamic because we're looking for small groups and big effects. And we're way ahead of recruitment, and we're very excited about that. But most importantly, from our standpoint, it demonstrates that we can leverage genomics and precision medicine to test novel agents quickly. We want to accelerate drug discovery. But an important lesson here for us, it's not just about having the ideas and the will and the patience. You need the ability to profile people in rapid fashion using an assay that can be done on a multi-center trial. And that took us almost a year to figure out. But once we've got this figured out, we think the lessons we've learned will allow the next trials in other diseases. We think MDS should be next, maybe lymphoma, to do this similar approaches, and we're excited about that. And I think what I'm most excited about was that a lot of people told us all these reasons why we couldn't do it. In the end, we just ignored that and went forward and did the trial anyway, and it's been incredibly successful to date. So with that, I'll just stop. I'll acknowledge the people in my group that did a lot of the work. Alan Shih has led all of our IDH TET work over the past 5 years. All of our clonal hematopoiesis work is done with this larger group with Mike and Kali and Amit and Richard and Kelly. Our IDH work comes out of long-standing collaboration with Craig Thompson and his lab and Ari Melnick and many others. And the LLS AML clinical trial is really led by Amy, and then John and Brian and I are right by her side to help her with everything. Thank you very much, and I'm happy if there's time to take questions.